Okay, good morning, everyone. I hope this day finds you well and that you had an enjoyable weekend. I hope you're keeping your spirits up. And yesterday was beautiful. I hope you were able to get outside a little bit and enjoy some of the sunshine out there. If you're able to hear me okay, would you please type yes into the chat window? Okay, thank you very much. All right, gang. So, as you know, today is your second lab exam on the muscles and tissue identification. Uh, Before midnight tonight, please log into the Class Marker website and complete that exam. If you do have a personal challenge of some sort that would prevent you from taking the exam today, please contact me about that through email and let me know what's going on. We can work it out so that maybe you can have some extra time to take that exam. But please log into Class Marker and take your second lab exam today. Jennifer asked a question, does spelling count? Yes, it absolutely does, because the computer is going to be doing the majority of grading, so you need to spell the terms properly. Other questions? For the test, do we need to be really specific? Well, yes, you do. You need to know exactly what the arrow is pointing to. Ashley is asking, can we use the word bank? Of course you can. There's no way that I could prevent you from doing it anyway, so of course you may use your word bank. Jennifer, you need to be very specific about the muscles. So if it's biceps brachii, you need to say biceps brachii. Anything else? Okay. All right, gang. So I will be monitoring email through today so that if something does come up, send me an email and we'll get it taken care of. Don't build up any anxiety about the actual process, but you do need to take your exam today. You have 40 minutes to take the online exam, which is a little bit longer than you would have been given in class. Anything else? Okay, gang. Stay focused. Make sure before you start taking that test that you don't have any distractions near you, right? Try to be isolated so that you can really focus on what you need to do for 40 minutes. Okay. So let's continue here. We were talking about chapter 10, which is the muscle physiology chapter. Jennifer is asking a question, should we use frontalis or occipitofrontalis frontal belly? You should use exactly what your word bank has, Jennifer. Okay. Let's continue into chapter 10 at this point. So bear with me. I'm going to share with you my screen and bring up a Word document. Okay. So at the end of last class, we were talking about, well, I believe at the end of last class, we had put up a flow chart in your notes where we talked about the pathway between muscle contraction and bone movement. And we started with the sarcomere shortening and went through a bunch of steps, including describing the connective tissue coverings around muscle that you find on page 298, figure 10.2. And finishing with the actual bone movement. So I believe that's the last thing we did. And so before I continue with new notes, do you have any questions about chapter 10 content so far? Feel free to type a question into the chat window. Feel free to unmute your microphone and ask if you have a question. Okay, I'm not seeing anything coming through the chat window, so I will assume that you're okay at this time. Gang, the next thing we're going to do is start talking about some microscopic anatomy of the muscle cells themselves. Okay, so let's see here. The next heading is going to be microscopic anatomy of skeletal muscle fiber. And remember, a skeletal muscle fiber is the same thing as a skeletal muscle cell, is the same thing as a myocyte. Okay. Each individual skeletal muscle cell of your body forms from the fusion of more than 100 smaller cells during embryonic development a very, very long time ago. Okay, these primitive cells from a long time ago are called mesodermal cells. Don't worry about that. That's not a big deal. That's a topic for an embryology course if you ever have a chance to take one. But at any rate, your individual muscle cells form from these primitive embryonic cells, which we call mesodermal cells. Okay, and that's the reason that your skeletal muscle fibers are actually multinucleated.
Dr. Allen, is that fiber or fiber for after muscle cell?
Thank you very much. That's a typo. Yeah, definitely fiber. Okay, each individual muscle cell forms from the fusion of more than 100 small mesodermal cells during embryonic development. And this explains why skeletal muscle cells are multinucleated. Okay. All right, we're going to take a look at some of the highlights of the skeletal muscle cells and put that into your notes. So perhaps page 298 in your textbook would be a good page to have open as we're talking about these things. I want to make you aware of some of the important anatomy that's relevant for muscle contraction physiology. First, we have sarcolemma. Okay. The sarcolemma is the fancy term we use to refer to the plasma membrane of a muscle cell. You know, of course, you might think to yourself, well, why can't we just refer to it as the plasma membrane? Well, as fields become more and more specialized, there's going to be terms that refer to specialized versions of things. So the sarcolemma is the plasma membrane of the muscle cell. Okay, you can see that highlighted sort of a light blue color on page 298. Okay, sarcoplasm. You can probably guess what that is. The sarcoplasm is the cytoplasm of a muscle cell. Okay. Now, I do want to point something out here within the sarcoplasm. The sarcoplasm of a muscle cell is going to contain an abundance of glycogen. Okay. So let's see here. So my next question, and I will type it in as a question here into your notes. Why do muscle cells have an abundance of glycogen? Can anybody answer that? Either type the answer into the chat window if you're If you want to talk today, go ahead and unmute your microphone. Why do muscle cells have an abundance of glycogen? I'm hoping that some of you can answer that. Okay, Pam says glycogen can be used for the synthesis of ATP. That's true. Okay, that's a very good statement, Pam. Can anybody else contribute to this conversation?
I was about to say to help the action potential.
To help the action potential. I appreciate you trying, but I don't think I can say yes to that one. Okay, anything else? Okay, so yeah, Pam was definitely in the right ballpark with this. Remember that skeletal muscle cells and liver cells are two of the major tissues that are going to respond to insulin in your body. So when you have excess blood sugar, the pancreas releases insulin, and then the liver and skeletal muscle cells, which we're talking about skeletal muscle cells right now, they will take in the extra glucose into the cytoplasm and link the glucose monomers, link the individual glucose molecules together into a chain of glucose called glycogen. And should the skeletal muscle cell need an extra reserve of biochemical energy to power muscle contraction, that glycogen can be converted back into glucose and then can be broken down for ATP synthesis through aerobic cellular respiration to power the muscle contraction process. Okay. Glycogen is a storage form of glucose. When necessary, the glycogen can be converted to glucose, which can then be used to make ATP necessary for muscle contraction. Okay, glycogen is a storage form of glucose. When necessary, the glycogen can be converted into glucose, which can then be used to make ATP necessary for muscle contraction. Okay, remember, in parentheses, remember, skeletal muscle responds to insulin by absorbing excess glucose from the blood plasma. Okay, gang. Does that make sense, what we're saying at this time? Are you okay right now? Okay, let's continue. One other point about the sarcoplasm that I feel that I need to note is that the sarcoplasm is going to contain a special protein called myoglobin. Okay. Now this myoglobin acts in a similar fashion to hemoglobin. Okay. Because it can bind oxygen molecules and ensure that the skeletal muscle cells have an abundant supply of oxygen to power aerobic cellular respiration for the purposes of making ATP to support the muscle contraction process. Okay. Okay. Inside muscle cells, we find myoglobin. This is similar to hemoglobin. Myoglobin can bind oxygen molecules. Okay, so I'm talking about O2 molecules. And the myoglobin inside the muscle tissue is going to act like an oxygen battery. That is, it's going to store the excess oxygen, and if the muscle cell needs the oxygen to power aerobic cellular respiration, then the myoglobin will release the oxygen so ATP synthesis can continue. I see your question, Lindsay. I'll answer that in just a moment. Myoglobin combine oxygen molecules and store it for when muscles need extra oxygen to power ATP synthesis through aerobic cellular respiration. Myoglobin is a compound, Lindsay. And what I mean by that, it's made of two different things. One of the things is the myopigment. So you're right, part of myoglobin is a pigment. The other thing is globin, which is actually a protein. So myoglobin is made of a pigment and a protein attached to each other. Okay, any other questions at this time? Okay, keep them coming. All right, number 4. Now let's talk about the myofibrils. Okay, the myofibrils are the contractile organelles of the muscle cells. Okay, these are the contractile organelles. You can see myofibrils listed on figure 10.2, page 298. They're listed in picture B there at the top, and perhaps a better designation of the myofibrils in picture C at the bottom. They look, in the drawing, they kind of look like cylindrical tubes, if you will. And you see the myofibril also on page 299. So take a look at a couple of these different pictures and make sure you know what I'm talking about when I refer to the myofibrils. These are the contractile organelles. Okay. The structure of the myofibril is what gives the skeletal muscle and actually cardiac muscle, if you look carefully, a striped or striated appearance. The structural makeup of the myofibrils is responsible for the striped/striated appearance of muscle tissue. Okay. And as one of your questions in your homework recently was talking about, you know, we talked about in our lab hour together, I think last week actually, or perhaps the week before, you know, when you work out at the gym and you do exercise and you lift weights, typically we get stronger, not because we're producing more muscle cells, but because we're increasing the number of myofibrils within each individual muscle cell. Okay. As you exercise, the number of myofibrils inside each cell increases, allowing you to become stronger. All right, gang, do you have any questions right now? Please feel free to type into the chat window. Okay, let's continue. Number 5. Number 5 is sarcoplasmic reticulum. Okay, the sarcoplasmic reticulum is the muscle-specific version of smooth endoplasmic reticulum. Okay, so you can expect it to do the same things that most smooth endoplasmic reticulum will do, which is to say to provide enzymes for a variety of chemical processes. However, the reason we're talking about it here is because in smooth muscle cells, in the muscle cells with the smooth endoplasmic reticulum in the sarcoplasmic reticulum, the sarcoplasmic reticulum actually stores calcium. So the sarcoplasmic reticulum acts as a storage depot for calcium, Ca+2, okay? That calcium will be released by a muscle action potential, which is something we need to talk about probably later today. We're going to start getting to this. So the calcium is released by an electrical signal called an action potential. And when the muscle generates an electric signal, it's doing so in response to a neurotransmitter that's been released from a neuron. Okay, so there's a whole chain of events that we need to talk about moving forward. Okay, so the calcium is released during a muscle action potential. Okay, and the calcium is important because when the calcium is released from the sarcoplasmic reticulum, it's the actual trigger that begins a muscle contraction, or that allows this whole sequence of chain of events to occur. Okay, so let me put it this way. The calcium +2 is released during a muscle action potential, which helps trigger the physical mechanism of muscle contraction. Okay. All right, so just a few more things here we need to talk about in terms of cellular anatomy. In addition to the sarcoplasmic reticulum, we have a variety of myofilaments. Okay. Now, myofilaments simply refer to chains of protein. You know, if you think about a tow truck that has a chain hanging off the back that you can use to hook up to a car and tow the car. That chain, that steel chain, is made of individual links that are interlocked with each other. One link is connected to another link, is connected to another link. Well, that's what you should be thinking of when you think about a myofilament. It's one link of protein connected to another link of protein connected to another link of protein. So these refer to chains of proteins. Okay. And the two that we need to think about are first the so-called thin filaments, which are actually made of chains of actin protein. Okay, made of actin protein. And we have thick filaments, which are made of myosin protein. Okay. So let's see here. All right. Trying to think. Okay, so you do see thick and thin filaments, the actin and myosin myofilaments, on the bottom right corner of page 298. Your textbook does point them out. And what I would want you to appreciate at this time is that these thick and thin filaments, these myosin and actin proteins, stack on top of each other, kind of like pancakes. They stack on top of each other to form part of a myofibril that's actually called the sarcomere. Okay, so the myosin and actin filaments stack on top of each other to form segments called sarcomeres. Okay. So I know if this is the first time you're viewing this content, that this can be overwhelming because there's so much new terminology, so much new cellular anatomy you have to think about. I promise you it gets better. You just got to spend some study time on this. Okay, so let's do this. Let's define a sarcomere. Gang, a sarcomere is a segment of a myofibril. It is a segment of a myofibril. All right. If you look on page 299 and you look at the myofibril in the center of the cell, you notice there's individual segments, and in that picture on page 299 there's a black bracket with the word sarcomere. So a sarcomere is a segment of a myofibril, and that sarcomere itself is made of overlapping thick and thin filaments, overlapping myosin and actin filaments stacked on top of each other. So number 7, sarcomere is a segment of a myofibril made of stacked myosin and actin filaments. Okay. Questions at this time? Is there anything about this cellular anatomy on page 298 or 299 that I can help clarify for you? Okay, all right, let's keep going then, gang. The next topic that I need to talk to you a little bit about is actually some of the muscle proteins that are important for this process. Now, there's lots of them, and I'm not going to type every one of them into your notes, but I am going to type a couple of them that I want you to pay particular attention to. So your book has a very nice table on page 302, table 10.2, page 302, page 302, that has a number of proteins in it, and I want to highlight just a few of those. Okay, so we'll start with the easy ones, things that we've already stated, and that is the myosin and actin proteins. So myosin, this is a contractile protein, okay? And actin, that's a contractile protein. What we mean by that, and which you'll see pretty soon, is that the actin and myosin actually mechanically move in relation to each other to allow for contraction to happen. Okay, so the myosin and actin are contractile proteins. Okay. Number 3, I'd like to point out tropomyosin and troponin. Tropomyosin and troponin are both regulatory proteins. Okay. So what we mean by that is literally the troponin and tropomyosin proteins regulate whether a muscle fiber can actually contract or not. And that's a process that we're going to explain in about 5 minutes, and we're likely going to take the rest of the class on that. So let's see here. If you want to see what troponin and tropomyosin kind of look like, you can find them on page 307 of your textbook, figure 10.7. Okay, figure 10.7, page 307. You do see an example of some actin proteins there, which are colored yellow. And then you see this thin yellow band of protein called tropomyosin. And you see those blue those three little blue balls there, which are the troponin, okay? So that's what I'm talking about in our conversation right now. Okay, yes, they're also on page 301. You may have stumbled there as well. Okay. And finally, I want to mention the titin and structural proteins. Titin protein is a structural protein, okay? I mean, in some sense, every protein we're listing is a structural protein because it's physically present as part of the structure of muscle cells. But we define titin and dystrophin as structural proteins, okay? The reality of it is that titin provides much of the elasticity of the muscle fiber. Excuse me. Trying to find a good picture for you here, but not quite coming up with a good one for the titin. Okay, titin is shown on page 300, figure 10.3. Page 300, figure 10.3 does have a picture of titin there, and you can see it has a coiled appearance, kind of like a spring, kind of like a, a metal spring. So that's titin. And then the second protein I want to at least put into your memory banks here is dystrophin. Okay. Dystrophin is the protein that links the end of a myofibril to the sarcolemma, and you can see it on page 299. Links the end of a myofibril to the sarcolemma. That is dystrophin. Okay, so we'll talk about it again as we go over this entire process. So we're going to trace it from start to finish. But I simply want to get these proteins in your notes so that you're at least aware of them, and that your teacher does think that these are important for you to know. Okay. All right, gang. Do you have any questions at this time? Okay, so Ashley has a question. Since smooth muscle isn't striated, does that mean it doesn't contain myofibrils, or is there some other reason? Good question, Ashley. The smooth muscle protein does contain myofibrils, but it doesn't contain as many of them. Because it doesn't contain as many of them, the overall appearance of the muscle tissue doesn't look striped. Other questions? Okay, gang. All right, so now we've gotten all that note-taking behind us. We have a background, a foundation to begin our conversation, which is understanding how skeletal muscle cells actually contract. And this is what we need to talk about. We need to talk about the physiology of skeletal muscle contraction. So, all right. What I think I will do next is to help you frame your thinking about this. I want to show a quick video to you, which I would normally show in class with you if we were together, to help you visualize this process so that when we're talking about it together, you can refer back to the memory of the video as to what we are talking about. So I'm going to show you a small video at this time and ask you to please do your best to stay focused and think about that video. And I will also send you the YouTube link to the video during this afternoon's email so you can watch it again during your own personal study time. Okay, so bear with me a moment, and I'll bring that video up. Okay. So on the screen right now, you should see a YouTube screen here with skeletal muscle contraction, high definition animation. Okay. So I'm going to let this play forward and let you watch it for the next 4 minutes. And certainly, if you have questions, jot them down during the video and you can ask immediately afterwards. So here we go, gang. Skeletal muscle contraction.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes. You
Okay, folks. So, there you had a quick overview of the process that we need to talk about in some considerable detail. So, do you have any questions about the video itself? Okay, do you have any questions about the video itself? Okay, all right. You folks are quiet today, that's fine. I hope you're okay. So what we're going to do is talk about the processes that were described in the video, and we're going to try to break them down step by step. So I'm going to try a mini experiment online with you here. I'm going to try to make some very basic drawings using a PowerPoint screen here. So bear with me just one moment. Okay. All right. So at this point, you should see a PowerPoint screen on your screen, and we're going to talk about the process of how neurons stimulate the skeletal muscle cells to contract. Okay, so in your textbook kind of describes the process of muscle contraction first, and then after that, it describes how the nervous system stimulates that muscle contraction. And certainly that's a fair way to approach it, but I'm going to start from the beginning, which is the nervous system doing what it does, then stimulating the muscle contraction. In other words, I'm not going to work backwards. I'm going to work forwards with you on this. So to explain this, you want to be looking at figure 10.9 on page 310. Page 310, figure 10.9. Okay. Okay, so let's see here. What we need is a neuron. Okay, so bear with me here. Okay, if you can see my horrible drawing on your computer screen, please type yes into the chat window. Okay, thank you very much. I just want to be sure that you're with me. Okay, so, all right, so we have a neuron here, and then I'm also going to draw the sarcolemma of a typical skeletal muscle cell. Okay, so this is the sarcolemma. Okay, and that means everything to the bottom of that line is actually going to be inside, right, inside the muscle cell. Okay, so a couple features of this anatomy that you should appreciate. Let's see here, let me get a different color here real quick. This structure here, as you can see from your own textbook, has a name. It's called the synaptic end bulb. Okay, the synaptic end bulb. The synaptic end bulb is going to be the location where neurotransmitter is ultimately released from the neuron, and we'll talk about that in just a moment. Okay, the distance between the neuron and the muscle cell is called the synaptic cleft. okay? So you have the end of the neuron, which is called the synaptic end bulb, and you have the space in between the neuron and the muscle cell, which is called the synaptic cleft. And the entire thing here is between the neuron and the muscle cell. Well, this is the neuromuscular junction. This is the neuromuscular junction. Okay? All right. So, at this point in our course, we haven't really talked in any great detail about what electrical signals are in the nervous system or in the muscle fibers. And that's going to end very soon because the next chapter we start is going to be a nervous system chapter. So we're going to be talking about that in great detail in the weeks ahead. But suffice it to say right now, when you talk about an electrical signal, in the nervous system, what are we referring to? I'm going to type that into the chat window. What is an electrical signal made of in the body? When we say electrical signal, what exactly is it? Can anybody answer that for us? Okay. So my question to you up top is, what is an electric signal? Okay, can anybody tell me what it's made of? What is an electric signal made of? Am I talking about electrons? What am I talking about? Okay, Pam says sodium. Lindsay says ions. Those are both pretty good answers, okay? And I can accept them right now, okay? An electric signal in biological systems, which of course is what our body is. Electrical signals in biology doesn't really refer to electrons that power the lights in your room. It refers to the flow of ions. Very good. The flow of ions, okay? So what's going to happen for the nervous system to stimulate muscle contraction? You know, think about the muscles in your arm and your forearm, your hand and wrist areas right now that are moving so that you can write notes out on a piece of paper, or you can move your fingers on a keyboard to type notes. You know, exactly how does that happen? Well, we'll go into the details of the nervous system business at another time, but what I will highlight at this time is that when you have an electrical signal occurring, okay, you're going to have these ions that are moving back and forth in a seesaw fashion across the plasma membrane of the neuron, okay? So as I'm putting these jigsaws in right now, you should envision tiny little ions, primarily at this point, sodium and potassium, moving back and forth across the plasma membrane of the neuron, okay? So that jigsaw pattern right now is the electric signal. Okay, and it is made of these ions that we hinted at, this flow of ions. Okay, that electrical signal is also known as a nerve impulse. Okay, that electrical signal is known as a nerve impulse, and a nerve impulse is also called an action potential. Excuse me. So we have this electrical signal, which is called a nerve impulse, which is known as an action potential. What that nerve impulse is going to do is it's going to, let me change my color here again. Okay, so embedded in the plasma membrane of the neuron are calcium channels. Okay, now they're not just any old random calcium channel, they're actually called voltage gated calcium channels. Voltage gated calcium channels. Okay. So let's see here. I'm going to try to write some basic notes here to kind of tell you what's happening in the drawing. Okay, and I will probably have to erase these notes at various points. But okay, so for the very first thing that has to happen for a neuromuscular junction to do its work, which is to stimulate a muscle fiber to contract, the very first thing that has to happen is that a nerve impulse has to spread down the neuron. And you're seeing it there with that little red jagged drawing there. So nerve impulse spreads to the end of the neuron, which you know is called the synaptic end bulb. Okay. When that nerve impulse spreads to the synaptic end bulb, it's going to encounter the voltage-gated calcium channels. As the nerve impulse is spreading along the plasma membrane, the voltage-gated calcium channels are going to change their shape in response to the nerve impulse. They're going to change their shape. And what's going to happen is, oh boy, let me try a different color here. I'll use this one. Okay, so sitting in the synaptic cleft is the synaptic fluid, right? The synaptic cleft is the space between the end bulb and the sarcolemma of the muscle cell. So there's a whole bunch of fluid here. Okay, there's going to be synaptic fluid. And in the synaptic fluid, there is going to be a whole bunch of calcium ions. Okay, so I'm just putting some dots here indicating calcium. So the nerve impulse spreads down the neuron, and when that nerve impulse reaches the voltage-gated calcium channels, the calcium channels change their shape, and the calcium actually enters through the channel into the neuron. Okay, so let me get a better color here for note taking. Okay, so number 2, after the neural impulse spreads to the end of the neuron, the voltage-gated calcium channels change shape, open, and they allow calcium to enter the neuron. All right, so there's a couple key ideas here we need to talk about. And first off, boy, I wish I was talking in front of you on a whiteboard. This would be so much easier. Okay, so another question coming through the chat window. What is voltage? What is voltage? Can anybody answer that? It might be one of those things where you could not have imagined that in biology you would have to know some basic principles of electrical engineering. But in fact, you do if you're going to understand. Okay, Voltage is a measure of electricity. That's a good starting point, Lindsay. I mean, that's an okay starting point. We can do a little bit better than that, but you're definitely in the game with that. What is voltage? Anybody else? Do we have anybody that's ever tinkered with electronics in any way? Okay. Any thoughts, gang? Okay. Well, just so we can move the conversation forward, let me tell you, voltage is a measure of the difference in electrical energy across the surface. Well, think about it. Over here, the surface is going to be the plasma membrane of the neuron. We have the inside where my cursor is now, and we have the outside where my cursor is now. So voltage is a measure of the difference in electrical energy on two sides of a membrane. A measure of the difference in electrical energy on two sides of a membrane. So what I'm trying to tell you is that the nerve impulse changes the voltage across the membrane of the neuron. The nerve impulse changes the voltage across the membrane of the neuron. Now that impacts the voltage-gated calcium channels, because what are the voltage-gated calcium channels made of? That's my next question. What are the voltage, coming through the chat window, what are the voltage-gated calcium channels made of? They're made of protein. Thank you, Jennifer. Jennifer, what are proteins made of? Okay, open question to everybody. What are proteins made of? No, they're not made of nucleic acid. Okay, Ashley G. Okay, Pam, good. They are made of amino acids. Proteins are made of amino acids, right? So this is where your foundational knowledge, the things that we wanted you to learn and master to some level in chapter 2 comes into play. Proteins are made of amino acids. And I won't take us back into the book, but you could go back into the book. And the point of it is that amino acids often have an electrical charge on them. Amino acids often have an electrical charge. And so what happens is the nerve impulse, which is the flow of ions, actually changes the electrical charge of some of the amino acids making up the voltage-gated calcium protein channels. And when their charge changes, their shape changes so that they open up. Right down here in the notes, I'm pointing out that the voltage-gated calcium channels change shape. Well, why do they change shape? They change shape because the amino acid charges are being neutralized momentarily by the ions moving back and forth of the nerve impulse. Let's see here. Okay. So this shape change that I'm highlighting on the board is in fact caused by the nerve impulse neutralizes charges on the amino acids, making up the voltage-gated calcium channel. Making up the channel proteins. Okay. All right. So once again, to get a muscle fiber to contract, the nervous system has to be involved. What happens is the nervous system sends an electrical signal, which we're calling a nerve impulse, down the neuron. This nerve impulse is really made of a flow of ions, sodium and potassium ions. Can anybody tell me what charge sodium and potassium ions have? What electric charge do sodium and potassium ions have? What electric charge do sodium and potassium ions have? Okay, +1, that's right. They have a positive charge. So you have these positively charged ions moving back and forth across the plasma membrane of the neuron. And when they encounter the amino acids of the calcium channel proteins that have negative charge, some of those ions bind to the amino acids of the channel and momentarily neutralize their charge. The end result of that is when you neutralize or change the charge on an amino acid in a protein, you are going to change the shape of that protein. So it's that shape change which allows this calcium channel to open up, and calcium ions can come through the channel and enter the neuron. Gang, question coming through the chat window. Two of them, actually. Are you with me? Do you understand? What can I try to clarify for at this time? What can I clarify or help you with at this time? You sure there's nothing at all that you would want me to help you with? All right. Okay. Well, let me know. Send me emails, write your questions down, bring them into future lectures. Okay. So continuing with our conversation, the electrical signal spreads down the neuron, changes the shape of the voltage-gated calcium channel proteins. Calcium enters the neuron. Okay. That's numbers 1 and 2 here in our notes. Okay, next. You'll notice from the drawings on page 310 that inside the synaptic end bulb, there's these tiny little bubble-like things, okay? And these bubble-like things are the synaptic vesicles. So inside the neurons, we have these bubbles, which are called synaptic vesicles. Okay, and the synaptic vesicles contain a very important cargo called, in this case, acetylcholine. Acetylcholine is the primary neurotransmitter that's going to be released at a neuromuscular junction. Acetylcholine is not just used at neuromuscular junctions, it's also used in the central nervous system for a variety of other purposes, but for the purpose of our conversation today, we're going to talk about it in the context of muscle physiology. Muscle stimulation physiology. Okay, so gang, I'm going to quickly run out of some room here. obviously you don't want to erase any of your notes, but I'm going to have to create some space here so that I can write some additional things. Okay, so bear with me just a moment. Ugh. See if I can find a way to erase some of this stuff. Okay. Okay, so remember, synaptic vesicles contain, generally speaking, neurotransmitter. Sorry, that's cramped. That's neurotransmitter. And for our purposes at the neuromuscular junction, the neurotransmitter you need to know is acetylcholine. Okay, so I will try to type in a little A character there inside the synaptic vesicles for acetylcholine. So when the calcium enters the synaptic end bulb, that triggers exocytosis of the synaptic vesicle content, okay? So we covered exocytosis a long time ago. I hope you remember what it is. It's a way of cells moving large cargo through their plasma membrane and pushing it out of the cell. So long story short, okay, these tiny little bubbles, they are literally going to move to the plasma membrane and they're going to push their cargo out into the synaptic fluid. Okay, so they come over here and they fuse with the plasma membrane. And the cargo is literally dumped into the synaptic fluid, okay? So those A letters at this time don't represent the letter A, they represent acetylcholine. Okay, so acetylcholine is dumped into the synaptic fluid. At that point, the synaptic fluid is going to be the medium, if you will, that the acetylcholine uses to get to the sarcolemma, because this content is going to move by simple diffusion over to the sarcolemma. Embedded in the sarcolemma that you can see on page 310 is actually acetylcholine receptors. So since I'm not too good at drawing, I'm just going to type in ACR for acetylcholine receptor. Okay, so you're going to have multiple acetylcholine receptors embedded in the plasma membrane of the muscle at this time. Okay, so question coming through the chat window. From Pam, question. So the synaptic vesicles will move to the plasma membrane and then the cargo will move to the synaptic fluid. Yeah, as soon as the exterior surface of the neuron plasma membrane ruptures, the acetylcholine will be dumped into the synaptic fluid. Yes, that is correct. Okay, so my question coming through the chat window, how many acetylcholine molecules bind to each acetylcholine receptor? How many acetylcholine molecules bind to each acetylcholine receptor? What do you think, gang? Okay, Pam says 2, and she's right. If you're not clear what we're talking about, take a look at picture C as in Charlie. Look at picture C on page 310 of your textbook. Do you see you have these little green acetylcholine receptor channels, and 2 molecules of acetylcholine are binding to each one in order to change their shape enough so that something can enter the muscle cell. So my next question is, what material enters the muscle cell when 2 acetylcholine molecules bind to their receptors? What material enters the muscle cell? Okay, so I'm going to keep writing some notes here. Okay, Ashley says sodium. Good job, Ashley. That's exactly correct. Okay, let me come over to the other side of the page here and write some basic notes. Okay, let's see here. Okay, so let's see what we have to put down is, after number 2, we had calcium entering the neuron. Okay, that triggers, at number 3, that triggers exocytosis of the acetylcholine from the neuron. Okay, so the calcium entering the neuron triggers the exocytosis of acetylcholine from the neuron. That acetylcholine will move by simple diffusion to the acetylcholine receptors embedded in the plasma membrane of the muscle cell, which is formerly known as the sarcolemma. So I'll just write it down as A, okay, meaning acetylcholine. A binds to the ACR, the acetylcholine receptors, okay? When 2 molecules bind to the acetylcholine receptors, the shape of the receptor changes, allowing sodium to enter the muscle cell. So when acetylcholine binds to the acetylcholine receptors, sodium ions enter the muscle cell. Okay? You may not know it yet. You may know it if you've had other courses, but you may not know it yet. When sodium enters the muscle cell, that is the very first step needed to depolarize the muscle cell. And depolarization is the very first step in an action potential, which is this thing up here, action potential. So the entry of sodium, and I'm watching our clock here, I know we're getting close on time. The entry of sodium triggers a muscle action potential. Okay, the entry of sodium triggers a muscle action potential, which is the same thing as a muscle impulse. Okay, a muscle impulse. Right, so, right, think of it this way. Here we have muscle impulse, which I'm highlighting in green, and up here we have nerve impulse, right? If the electrical signal is happening in a neuron, we call it a nerve impulse. If the electrical signal is happening in a muscle cell, we call it a muscle impulse. So it's some form of impulse, and which form it is depends on what kind of cell the activity is occurring in. All right. Looks like we're just about out of time, so let me ask you, do you have questions at this time? Okay. I'm going to, well, okay, maybe I should give you a little bit more time to finish your drawings there, finish your note taking. Gang, we're about to stop our time together since we're out of time. Do you have any questions right now about the content or about taking your exam today? Remember, you must take your muscle exam online today by midnight. If you do have, if you do have a personal circumstance occurring that would make it hard for you to take your exam today, contact me through email and we'll work something out. How many questions are on the exam? 35. 35. Okay, question. Let's say you pointed biceps brachii. Do you want us to say biceps brachii or be specific and say the long head or short head? Okay, because I did not have long or short head on your word bank, you don't need to use long or short head terminology. Simply say biceps brachii, Jennifer. Okay. While the computer is going to be grading these exams, I will be looking them over. So if there's something very silly about how the computer graded something, I will correct that. Okay, so don't worry too much about that. Okay, gang, I'm going to stop sharing my screen and just go back to you so that you can see me. Is there anything else you'd like to ask questions about before we end here? Okay. I will be remaining online for a few minutes. If you have something about course content you want to ask about, I'll be online, or you can send me an email. Otherwise, I would ask that you please, sometime today, while you're fresh and you're free of distractions, please take that lab exam 2. It's going to take me a couple days to get all of that wrapped up and finished for grading purposes. But when I do have them finished, I will let you know so that you can log in and get your score. Okay, any final questions, gang? All right. Then I'll say goodbye to you at this time and wish you well. Please stay safe out there. Things are getting very, very serious, so use good sense when you're going outside. And I'll see you all on Wednesday. Okay, now. Okay, hi folks. I'm back on. If there's anything I can help you with, please let me know.